is Hye-won Seo. And could you give me some time to share my PPT first?
Sure, yeah.
Okay, and can you guys see the PPT well?
Yes.
Okay, great. And okay, awesome. Before I start, thank you for this opportunity and time for this presentation to all of you guys. My name is Hye-won Seo. I'm currently working as a postdoc in UW-Madison. And if you guys have any question while I'm talking, please interrupt me whenever you guys want. Uh, this is a slide about me. I'm from South Korea, so I can speak in Korean language and English. And I like listening to music, chatting with friends, and working out at home. And in science aspects, I've always interested in the virulence of infectious microorganisms, and I got bachelor's degree in food nutrition and biology. So I wanted to study the effects of nutrition and virulence of pathogens at first. So I contacted Professor Cheon Yoon, who works on the effects of micronutrients on infectious microorganisms. After I got PhD, I contacted Nancy Keller, who works on secondary metabolites from microorganisms, including Aspergillus mycotis. So now I'm working as a postdoc in a lab, and this is my second year. Through whole this journey, I've got these experimental skills, as you can see, and now I'd like to talk about my previous and current works. Well, I've worked on various microorganisms like yeast and bacteria, but I majorly worked on Aspergillus mycotis, which is a species of fungi. These main habitats are soil, decaying vegetation, and mammalian lung cells. Aspergillus mycotis is really common, so we usually inhale from 500 to 5,000 spores every day, and these spores can reach to the lung cells. Aspergillus mycotis can be eliminated by the host immune cells. However, if host immunity level is abnormally too low in like immunocompromised individuals or those with pre-existing lung conditions, uh, then not eliminated fungus forms their hypha in the lung cells, making the host difficult to breathe. This is called aspergillosis with high motility rate. Factors that Factors that help Aspergillus mycotis settle down and survive in the host are called virulence factors, impacting on adhesion ability, or thermotolerance, nutrient acquisition, cell wall integrities, stress response, hypergrowth, and interaction with the host immune cells. And many secondary metabolites from Aspergillus mycotis are known to be involved in virulence factors. Secondary metabolites are organic complexes which are not directly involved in basic cellular activities, but synthesized for various purposes like development, defense, nutrition acquisition, and interaction with other microorganisms. Usually, these genes involved in biosynthesis of specific secondary metabolites are arranged sequentially on chromosome, which is called biosynthetic gene cluster, and I'm going to say BGC. And it contains core enzymes and tailoring enzymes. Core enzyme is an enzyme making a big basic scaffold of the secondary metabolite, and tailoring enzymes modifies the structures of intermediates such as oxidases, hydroxylases, or methyltransferases like this. The expressions of genes in BGCs are regulated by transcription factors. I'm going to say this TF. And there are two types of TFs. One is global TF, and the other one is pathway-specific TF. And I'm going to say this PSTF. Global TF is a factor regulated by external environmental stimuli, and the PSTF is a factor regulating the specific BGC's expression. The most PSTFs are located in the BGCs, but some BGCs don't have PSTF in their cluster. It was predicted that Aspergillus megatus harbors like 34 to 36 BGCs in total, but only 20 BGCs were defined, and the rest of BGCs are still unknown. So scientists have tried various reports to define unknown BGCs, including genome mining, changes in environment factors, activating or deactivating some BGCs, and co-culturing with other more molecular organisms. Through these trials, they could also find out that many BGCs could interact with each other. The topic of my PhD project was similar with this. Although I worked on known secondary metabolite BGCs, I could find out two different BGCs are connected each other. So through PhD course, I revealed that zinc induces different expression patterns of gliotoxin BGC and Chardonnay flagellin BGC. Gliotoxin BGC is called Gli-BGC, including their own PSTF, Gli-Z, in their cluster. Chardonnay and flagellin's cluster, flagellin BGC is called Puzo BGC and FMA BGC, respectively. Interestingly, these two BGCs have unique structures. As you can see, greenish, brownish genes represent the Puzo genes, and the black genes represent FMA genes. And as you can see that these two BGCs are intertwined and overlapped. In addition, they share a common PSTF in their clusters, which name is FUME-R, or other labs called it FAB-R, but I'm going to just say that FUME-R, including inducing Puzo BGC and FMA BGC at the same time. So first, I want to find out the impact of zinc on secondary metabolites. So I cultured wild-type Aspergillus megatus in the media with different zinc concentration, and I extracted the secondary metabolite from the supernatant and checked the production of secondary metabolites through HPLC. And I could find out while increased zinc inhibited gliotoxin production, it activated production of shortin A and flagellin. As these secondary metabolites are representative toxic secondary metabolites, it was interesting that zinc concentration could govern the expressions of these secondary metabolites. As I wanted to check if zinc affects on RNA expressions of Gli-BGCs and Puzo FMA BGC, wild type of Aspergillus megatus was cultured in the media with different zinc concentration, and RNAs were extracted from the mycelia, and the northern blot was proceeded. First, I could find out that higher zinc concentration inhibited Gli gene expression, including PSTF Gli-Z. On the other hand, higher zinc concentration activated RNA expressions of Puzo and FMA-PKS and FUME-R, which is a PSTF of Puzo and FMA BGC. This northern data align with the previous HPLC data. So I want to see that if ZAF-A, which is a global TF induced by low zinc, is related in this phenotype. So I generated ZAF-A deletion and ZAF-A complemented strains of Aspergillus megatus and cultured these strains with wild type in the media without zinc. RNAs were extracted from mycelia and gliotoxin was extracted from supernatant. When ZAF-A was deleted, RNA expressions of Gli genes and gliotoxin production was abolished. However, when ZAF-A was complemented, the RNA expressions of Gli genes were recovered and the production was also increased than wild type, which means ZAF-A promotes gliotoxin production. As you can see, ZAF-A impacts on the expressions of Gli-Z PSTF. I want to check if ZAF-A binds to the promoter of Gli-Z. In the previous other paper, the binding motif of ZAF-A was reported, and I could find that there are two sites of ZAF-A motifs in the Gli-Z promoter. So I made 3 types of mutants like this which the motif sites were points mutated in Aspergillus megatis, and checked the gliotoxin production from these mutants. Although the pattern of gliotoxin of M1 mutant was not consistent and unstable, gliotoxin production of M1 and M1, M2 mutants were decreased than wild type. I also proved that the direct binding of sulfate protein to the Gliese promoter through electrophoretic mobility shift assay. 100 base pairs of fragments of wild type promoter was labeled with P32, and wild type and each mutant's promoters were used as unlabeled competitors. So I could see that competitors of cold wild type induced a lot of sulfate unbound labeled probe, but the competitors of mutants could not induce sulfate unbound labeled probe, which means only wild type fragments binds well with sulfate protein. However, in case of fume R, when sulfate was deleted, the RNA expressions of fume R and shortening production was increased than wild type. When sulfate was complemented, the RNA expression of fume R and shortening production was recovered as wild type. The thing is, in the promoter of fume R, there is no binding site of sulf-A, which means sulf-A inhibits production of shorten A by activating expression of fume R without directly binding to its promoter. Interestingly, in the low zinc conditions, when Gliese was deleted in the fumigatus, shortening and fumagillin products were increased than wild type, and deletion of Gliese induced high expressions fume R, which means Gliese, which is a PSTF of Gli-BGC, inhibits production of shorten A and fumagillin by activating expression of fume R. So I wanted to check if the inhibiting fuso FMA BGC of sulf A is mediated by Gliese. So I generate Gliese overexpression strains in the background of sulf A deletion strain by adding thiamine promoter. Interestingly, although sulf A was deleted, overexpression Gliese induced gliotoxin production as wild type and less production of shorten A than wild type, which means that inhibition of shorten A by itself A is entirely mediated by Gliese. As Gli-BGC and Puso FMA BGCs are linked, I wanted to see if gliotoxin, shorten A, and fumagillin has synergistic effect in virulence. I induced neutrophil and macrophage from HL60 cells and infected Gliese mutant, FUMAR mutant, and Gliese FUMAR double mutants of Aspergillus fumigatus to the neutrophil and macrophage and checked the survival ratio of these fumigatus strains. And I could see the survival ratio of double mutants were similar with single mutants. I also tested the virulence of these strains with mice. I infected Aspergillus fumigatus strains to mice through nasal infection and checked the survival rate of mice. The survival rate of mice infected with Gliese mutant and Gliese FUMAR double mutants were similar, which means their products do not have synergistic effect, but Gliese-BGC and Puso FMA BGCs are related to virulence, respectively. So I found this mechanism from Chota News Lab, and I've got most of scientific skills which I have from the PhD course, including molecular genetic works, chemical works, and mouse works. And I was excited to see the interaction between different toxic secondary metabolites. So I contacted and applied for Nancy Keller's lab as a postdoc to work other interactions of secondary metabolites. Interestingly, they already developed a computer program which can predict the connections of known or unknown transcription factors and secondary metabolites. The name of this program is GRASP, the abbreviation of the gene regulation of Aspergillus fumigatus. Nancy wanted me to check if the models from GRASP are correct and define the detailed mechanisms of the regulation. For my project, I choose three models from GRASP, and this is one of the models. Big red dots represent genes encoding transcription factors, and small blue dots represent normal genes. This model is predicting that transcription factor encoded by 3G11990, which is centered in the round, and HSFA, which is a heat shock factor A, might be related with gliotoxin production. First, we named this TF encoded by 3G11990 as a regulator of gliotoxin, which is ROG-A. And this ROG-A TF is a novel transcription factor. And HSFA is, as I mentioned, is a heat shock factor A, which was already reported as important for fungal growth. But the relationship between the secondary metabolite and HSFA was not reported yet. In addition, as you can see, these two transcription TFs are linked with GLI-T and GTMA. These two genes are known as genes breaking the disulfide bridge in the gliotoxin part, leading detoxifying gliotoxin. Because of the disulfide bridge, gliotoxin is toxic even to Aspergillus mycotis, although mycotis produce the gliotoxin. And this model suggests that these transcription factors are not only related with gliotoxin production, but also related with self-protection against the gliotoxin. So I generated ROG-A mutant and HSFA mutant respectively. As HSFA is lethal gene, knockdown HSFA strain was generated by adding xylose promoter in front of HSFA. And I cultured these strains and checked the gliotoxin and gliotoxin derivative bimethylgliotoxin through LCMS. And as you can see that ROG-A deletion and knockdown HSFA induce more gliotoxin and bimethylgliotoxin, whereas overexpression ROG-A and overexpression HSFA reduce gliotoxin and its derivatives, which means ROG-A and HSFA inhibit gliotoxin production respectively. And I also checked the RNA expressions of GLI-BGC's genes. And when you can see that overexpression ROG-A and overexpression HSFA inhibited the most of the GLI genes. The interesting point is when you can see the overexpression ROG-A, GLI-Z was totally downregulated, but overexpression HSFA didn't affect the GLI-Z expression, which means usually GLI genes are known as regulated by PSTF GLI-Z. This mechanism is really new, could be new thing regulating GLI-Z, especially working with HSFA. So first, I want to see that how GLI-Z is important in the regulation. So I overexpressed GLI-Z in the background of overexpression ROG-A and overexpression of HSFA respectively, and check the gliotoxin production. Then you can see the panel A, overexpression GLI-Z in the background of overexpression ROG-A was similar with the single overexpression GLI-Z. However, overexpression HSFA and overexpression GLI-Z was highly produced gliotoxin than wild type, but it's still less than overexpression GLI-Z, which means ROG-A dependent gliotoxin regulation proceeds through GLI-Z, but HSFA dependent gliotoxin regulation is not entirely mediated by GLI-Z. So, which means ROG-A regulation and HSFA regulation are distinct pathways, and I want to check if those two pathways interact with each other. So I checked the HSFA expression in the ROG-A mutants and ROG-A expression in the HSFA mutants. When you see pattern A, ROG-A didn't affect HSFA expression, but when you see the HSFA mutants, HSFA induced ROG-A. So HSFA positively regulate ROG-A expressions. So ROG-A regulates GLZ and HSFA regulates ROG-A, but the inhibition of HSFA on gliotoxin is not entirely relies on GLZ. So I want to figure out how ROG-A is important in HSFA's inhibition of gliotoxin production. So I generated a ROG-A deletion strain in the background of overexpression at HSFA, and I observed the gliotoxin and biometric gliotoxin from these strains. And it was found out that although HSFA was overexpressed, deletion of ROG-A increased the gliotoxin and its derivative production than overexpression HSFA. But still, it is less than wild-type production, which means HSFA inhibits gliotoxin production through two pathways. One is mediated by ROG-A, and GLZ is also mediated too, and the other one is ROG-A is not mediated. And when you can see the model, ROG-A and HSFA might be related with a self-protection against exogenous gliotoxin. So I cultured these ROG-A mutants and HSFA mutants under the solid media with some gliotoxin and checked the diameter and compared the diameter with the untreated wild type conditions. And when you can see that ROG-A deletion strain was more susceptible to the exogenous gliotoxin, and the overexpression ROG-A recovered its resistance. And when you can see the overexpression HSFA, when a higher HSFA induced more resistance of exogenous gliotoxin. So gliotoxin resistance is dependent on ROG-A and HSFA respectively. Not only this model, I also working with other two models, and as you can see, this model is suggesting that SKN7 and other transcript factor encoded by 1G 14945 are predicted to be linked to production of hydrofolic acid and formitrimorgan. I'm still working on the molecular work, but briefly, I can figure out that SKN7 is a negative regulator of hydrofolic acid, whereas 1G 14945 transcript factor is a positive. Is also a regulator of formitrimorgan C. And this is another model suggesting that NSDD and the hexa dihydroastochrome and xanthocillin production. I'm still working on the xanthocillin, so I can just show this data about Has. And tyrosine D is a precursor of hexahydroastochrome because this molecule is too big to detect HPLC or LCMS. So we can see that overexpression deletion NSDD produced more tyrosine D, whereas overexpression NSDD was recovered as wild type, which means NSDD negatively regulates the production of tyrosine D. So I'm wrapping up this project and I'm writing the manuscript about this project, and I'm planning to submit this paper maybe this month or early next month. And I got a chance to write a review paper before from JOF. So we got a lot of changes. So now it's not a review paper, but still it deals with the timeline of identification of secondary metabolite BGCs in Aspergillus nigotis. And I got honored to be chosen as an issue cover. So yeah, thank you for your time and yeah.
Thank you, Hye-won. Yes. Any questions from the panel for Hye-won?
Thank you for
David,
the time,
you
guys.
moved.
I have a few questions just to get some context since I don't work in the fungal pathogenesis and virulence areas. So it seems like there are a lot of secondary metabolites that some of which are involved in the ability of Aspergillus to cause disease.
Yes.
And gliotoxin, its name implies it's a toxin. What does it do to host cells itself? Is that known?
I want to show you.
Or any of these others,
Oh.
are they understood to sort of how they enable virulence?
In the gliotoxin structure, there's a disulfide bridge, and
Yeah.
disulfide bridge has an open electron region, and this region is really reactive, so it can reduce the ROS in the host
Okay.
human cells.
Okay, so it triggers ROS type response
Yes.
In the host.
Yes.
And it seems like that there are a host of these types of things that Aspergillus uses. So yeah, it's very complex to sort them out. The zinc effects that you initially started working on, is that thought to be because zinc is in low abundance or is in low availability in the host context? Is it known if any of these transcription factors or other regulators themselves bind zinc to measure zinc concentrations? Yeah.
Yes. Actually, zinc is the only one of the global transcription factors, and
Yeah.
there is other transcription factor was reported that it could be related to the South A. So
Yeah.
which name is RGLT.
Yeah.
Yeah, so it could be the one of the other ultimate upper transcription factor, I think.
But do any of these have zinc binding domains and the like that directly?
No.
So
No,
there's
no.
some other detection mechanism in the cell for
Yes.
reading out zinc concentrations that then
Yes.
trigger these other, okay. And are there other things known besides zinc concentrations that in environmental factors that might be present in the host that trigger or set up this network of responses?
Could you say
Yeah,
one more time about the
sorry.
question?
Yeah, well,
Sorry.
environmental factors beyond zinc that regulate or affect the expression of these metabolites that might be relevant for virulence.
There's a lot of other external stimulants exist. Yeah. So there could be light, there could be nitrogen, or there could be carbon, or pH, temperature kind of things. All of things are related in inducing the global transfer factors.
Okay, so those have been studied by various folks in the field.
Yeah, and although I'm only focused on the zinc nutrition, but it is already known that most of global transcription factors are related in the secondary metabolites.
Yeah.
For example, LA is a light sensitive transcription factor, and it governs the most of the secondary metabolites in Aspergillus mycotis.
Okay. Thanks. Nice work.
Thank you.
So light is a sunlight or specific wavelength. So I'm not in this area. Hope my question's not stupid.
Yeah. I think it's just sunlight. So when we induce the lead air, we culture those things in the dark room.
Thank you.
I have just one question. That is more a curiosity. The gliotoxin that is secreted by the fumigatus, right? Aspergillus. Do you know anybody working on the effect of gliotoxin on a bacteria like staph, by example? Just like how it's get in and what it will do?
Usually effect of gliotoxin to other microbacterial is really well known, and it is already known that gliotoxin represents antibacterial things and antibiotics like that. And usually, although Aspergillus mycosis have their own transporter, and it could be penetrated through the membrane to the bacteria.
Okay, so it just going through the membrane like that. Is that almost like the same type of mechanism like the thiocyanin of Pseudomonas?
Yes.
And then did you hear any instance of the bacteria become resistant to gliotoxin? Like efflux pump, by example?
Actually, sorry, I don't remember the example of that, but usually, yeah, gliotoxin resistance. Oh, sorry, I don't know about the gliotoxin and bacteria.
Yeah, that's just a curiosity. Yeah, because I just wonder because if they cohabit with other bacteria, and then so the bacteria can develop some kind of resistance against, just like bacteria always do. They survive by fighting back, and I'm just curious.
Yeah, makes sense. You're right. Yeah, usually bacteria get resistant to the antibiotics, and if the gliotoxin has an antibiotic traits, then yeah, it could be resistant too.
Okay, thank you.
So I have a question. Yeah. So I can see from your application dossier. So you did some homework to understand the research here. So my question is, what has motivated you to apply for this position? And how would you contribute to expertise to the research here?
Oh, well, I think the ultimate goal of studying virulence of pathogen is to develop the effective treatments of pathogens. And although I enjoyed my research, my research has been focused on the genetic regulation. So I want to expand my scientific scope into more practical area. So I was searching the labs focusing on the antibiotic resistance or antibiotic development, and now so I was glad to see that you guys are working on the combination of blue light and antibiotics. So, and I saw the papers published in your lab, and I thought I could do the experiments most of you guys done. So that's why I applied for this job.
Thank you. Yeah. Any questions from other people? Yeah.
Hi, Hye-won.
Hello.
Nice to meet you. Great presentation.
Thank you.
Yeah, I'm also interested to know more about you. How do you think, because now you are changing for a new field, if you come here to work with us, you'll be working with microorganisms, antimicrobial therapy, but like a light-based antimicrobial therapy now. And we are also working with translational studies. I want to know from you, how do you think this will help you in your future career?
My future career? As I told you, my career has been focused on the genetic regulation of secondary metabolites, but I want to apply this knowledge to the development of developing treatment methodology. So the working experiences in your lab will support me I'm interested in the development of antimicrobials, and I think developing photosynthesizers from microbials like gliotoxin kind of thing could be the good candidates of photosynthesizers, I think. So I can apply this knowledge about my secondary metabolites to the light therapy protocol, maybe.
Any more questions? So yeah, so if there is no more questions, so I would like to wrap up the interview today. Well, thank you again, Hye-won, for your presentation, also your interest in this position. So we will be in touch with you regarding the next step.
Great. Awesome. Thank you.
Thank you.
Okay, thank you.
Thank you, guys.
Mhm.
Bye.